Joachim Wegener is a full professor for bioanalysis at the University of Regensburg at the Institute of Analytical Chemistry. And this will be a talk where he used the Atlas system, one of our impedance-based systems. So I'm looking very much forward to it, where he will investigate the pharmacology of G-protein coupled receptors.
So I'm happy to be here. And in one way, it is really a perfect match that I'm the final speaker today, because I realize that I'm clearly an alien here. The alien because you are all interested in proteins that transport charges, ion channels, transporters. Somehow they mediate charge transfer across the membrane, internal membrane, plasma membrane, but we are not. We are interested in G-protein coupled receptors, so another class of cell surface receptors, which are not mediating charge transfer across the membrane. But still, we are using electrochemical technique to profile their pharmacology. And we are based on impedance technique, as you just pointed out, and that's what I would like to share with you today. But for those of you who may not be too familiar with GPCRs, let me give you the GPCR story in a nutshell. So GPCRs are seven transmembrane helix receptors in the membrane, and they are the largest protein superfamily that we know about. So we have about 800 genes in the human genome, which are encoding GPCRs. So no surprise that they are extremely important in human physiology and also in pathology. So we meet them everywhere, and that's why I said up here, it's one of the most relevant drug targets. When you look at the ligands that bind to these receptors from a chemical perspective, you realize there's almost anything there that you can think of, from small molecules to big proteins, ranging from hormones, pheromones, neurotransmitters, odorants. So it's an enormously wide spectrum of ligands. And there's this one figure that everybody who works in the field of GPCRs refers to sort of underline their importance, and that's the figure down here, that about more than 40% of prescription pharmaceuticals today, they do address GPCRs. Now, as I said earlier, they do not mediate charge transfer. So what they do is they have an extracellular binding site, and the ligand binds to this binding site, and it induces a conformational change of the protein. And as a result, here on the example of the adrenaline receptor, what happens then, once the ligand has bound and there has this conformational change going on, a G protein will bind to this receptor, G protein coupled receptor, and this G protein gets activated upon binding to the intracellular domains of the receptor, and then itself, it will activate some other enzyme. In this particular case, talking about the adrenaline receptor, it is adenylate cyclase, a membrane-bound enzyme, and it will produce cyclic AMP as a second messenger. And this second messenger will then sort of diffuse into the cytoplasm, and it will trigger all kinds of physiological processes. It's like an avalanche that gets triggered. Now, how can you measure now the activity of these receptors if there is no charge transfer across the membrane? Well, what people do is several ways. One way is that I better use these ones here, that they use agonists or antagonists, and you see the star here, radio-labeled ones, and then they do radioligand binding studies. But what you get from these is only the binding constant, only in quotation marks, but you don't get any information about the real activation of the receptor. If you would like to do functional assays and learn about whether the receptor has gotten activated, what you can do is you can measure the G protein activation. There are ways to do so, or you can measure the cyclic AMP here as a representative of the second messengers. So these are the typical ways that are pursued in order to study functionally how these receptors are operating. What we do is something different. We do cell shape changes. So we are measuring the change in cell shape with the help of impedance technique in order to profile the pharmacology. And I would like to compare this technology here with the other three by showing you this analogy here. Oh, sorry, no, there was it. So instead of looking into any compound of a car, like the clutch or the brake or the shear, what we do is we are looking at the car while it is driving. So we let the cells sit on our electrodes and we stimulate the receptor, and then we see what happens in terms of cell shape change. With the other assays, measuring calcium, measuring cyclic AMP, measuring G protein activation, it's more like looking on one of these parts of the car to understand how it works. So it's a very different approach. It's a holistic, it's a very integral approach because it integrates over the entire cell body. But in return, it doesn't rely on any reagents, any dyes, whatever. Yeah? It's a completely label-free approach. And there is a famous pharmacologist in Germany, and that is Ethel Costenis. And she said the approach here that you analyze individual components means that you only see what you have planned to see, but you miss the whole rest because you have to decide ahead of time whether you would like to measure cyclic AMP or G protein activation. So you sort of have kind of a bias on your experiment. When you do the impedance-based assay, it's different. She also has a quote for this. You will see everything, but you don't know what it means. And I would like to show you today that this is maybe not quite true. You can know what it means, and you can make use of these very integral data that you get. Now, how are we going to measure the cell shape change? Of course, we use nanion technology. That's why I'm here. So what we have here, you look into the wells of a 96-well plate, and on the bottom of the well, there are gold film electrodes, nothing more than gold film electrodes. We grow the cells on the surface of these electrodes, and then we measure the impedance of this system here. So the medium is on top of the cells. Typically, one electrode is rather small, the other one is rather big, and thereby we make sure that the impedance that we are reading comes from the small electrode. As the impedance scales to the inverse of the area, so the smaller the area, the bigger is the impedance, and when one is small, one is big. The impedance is dominated by what happens here on the small electrode. And now we measure this impedance, and from these impedance readings, we can measure cell shape changes. I will show you in a second. But first, we have misused the CardioExcite to start with. We used the CardioExcite system in order to do this GPCR work. And we used the 96-well plates, and we put them here on this tabletop device, and we measured our signals there. Or lately, we have been turning our heads towards the Atlas system because it has much higher throughput, and we can do like 6 96-well plates at the same time. But we have just been starting to using this machine. Now, why do we see cell shape changes from impedance readings? And the answer is when you look at the electrode from the side. So what you see here is supposed to be the gold electrode. Here is some insulating film that delineates the shape of the electrode. And when you look at the current flowing out of this electrode into the electrolyte, it's pretty much not impeded when there are no cell presence. But when cells are present, this changes because the cells behave pretty much like insulating bodies if you go from kilohertz regime to lower frequency. The membrane is pretty much insulating, and because it is insulating, it limits the current flow. And when you look closer, you can see that actually, because of the insulating properties of the membrane, the current has to flow around the cells. It has to pass through the narrow spaces underneath the cells and between the cells, and that makes it sensitive for cell shape changes. So whenever the cell changes its shape, it means that these current pathways will change. And when they change, the overall impedance changes. And the good news is that that is very sensitive. It's way more sensitive than the resolution of a regular optical microscope. And that makes it so versatile to work with. Now, what is the typical workflow that we are using for our impedance work, for our GPCR work? We start out with a cell-free electrode, and we seed our cells. Typically, we seed them to confluence, or we seed them such that they are confluent after 48 hours to allow for proper receptor expression. And you can follow this phase by just following the impedance, because when the cells attach and spread on the electrode, they block the electrode, impedance goes up. You will reach a new equilibrium, which is now the confluent cell layer. And then we start with our experiments. Then we stimulate the receptor by one way or another. And then we can follow how the cell responds to the stimulus by looking into the impedance, which reports on cell shape changes. Now, let me show you this system first on the example of the beta-adrenergic receptor. And in this example, we have taken primary cultured cells from bovine aorta, and we have grown them on the electrodes. You see here an electrode covered with cells. In this particular case, it's probably like 50, 60, 70 cells. And what is important to me is these cells have an endogenous receptor density. They are not engineered. They are just with endogenous receptor density on the surface. And of course, endothelial cells, they do express the beta-adrenergic receptor. So they are our prime target here. So once we had grown them on our electrodes, we then did the experiment. And what you see here is then the impedance as a function of time. And you see when we stimulated the cells here with 10 micromolar isoprenaline, which is simply a synthetic derivative of adrenaline. And as you can see from the control here, we add the isoprenaline, and you can see how the cell shape change that is induced by the signaling cascade essentially leads to this increase in impedance. So here we have the case that the beta-adrenergic receptors is coupling to the G-alpha protein with an S. S stands for stimulatory, because it stimulates adenylate cyclase, it increases cyclic AMP, and then the avalanche breaks loose. And this avalanche leads essentially to the cell shape change that we are seeing here. For the control, no isoprenaline present, we don't see any cell shape change. Now, what can we do with this? Of course, pharmacology means always quantitative approaches. So we take the change in impedance for a given concentration of isoprenaline, and we repeat the experiment for several concentrations and get dose response curve. And this dose response curve, simply by taking the change in impedance as a function of isoprenaline, allows us to calculate an EC50, which turned out to be 0.3 micromole. We compared it with a KD, so with a binding constant of isoprenaline, radio-labeled isoprenaline, to this particular cells, and found it to be very, very close. The KD was 0.2 micromolar here. Within error limits, we get the same answer. Now, but the question, of course, is, referring back to the quote that I gave you earlier, do you really know what you're seeing? Is this a specific response? So what we did is we went into antagonist mode. Instead of just using an agonist, as we did here with the isoprenaline, we pre-incubated the cells with 10 micromolar alpranolol. Alpranolol, for those of you who know what the adrenergic receptor, is a beta blocker. It specifically binds to the adrenergic receptor, but it blocks it. It does not activate the receptor. And that's what you can see. When we add the alpranolol, there is no response of the cell because the signaling cascade is not induced. And if we add 1 micromolar isoprenaline, there's no response either, simply because the antagonist is blocking the receptor. And here, if you remember, the EC50 was 0.3 micromolar, so we are 3 times above EC50. We should see a clear response, but there's none. So it's a specific response that we are seeing here that is induced by the beta agonist, and it leads to cell shape change that is picked up here. Now, there is several types of GPCRs. I told you about 800 genes, and they do couple to different G proteins. I was referring to this example here. The beta-adrenergic receptor binds to G-alpha S, stimulating adenylate cyclase, producing cyclic AMP. But there is others that bind to a G protein, which is G-alpha I. It's inhibitory. It inhibits adenylate cyclase, reduces cyclic AMP. And of course, there are these ones, which are coupled to G-alpha Q. G-alpha Q has a very different signaling pathway. It eventually leads to the activation of phospholipase C, and it then leads to the mobilization of calcium and diacylglycerol. So different second messengers are produced or reduced in concentration by different G proteins that are involved. I will really focus here on these 3 major pathways. There are others for experts here in the audience, but I will focus on these. And we look in all of these, and here is basically the summary. So this is what I've shown you before. This is the beta-adrenergic receptor. You see the isoprenaline here. G-alpha S, adenylate cyclase, cyclic AMP goes up. We can see the response, and we can do antagonist studies as well, and we see no response whatsoever. Here, we use the cell line that endogenously expresses the histamine 1 receptor, which is G-alpha Q coupled. And as you can see here, when we stimulate it with histamine, we get this clear response. If we have an antagonist ahead of time, mepyramine, adding the mepyramine here, we see no response, and adding histamine here as well, the response is not there. So it's a specific signal. And eventually here, we have a representative of the receptors that couple to G-alpha I, which is in this case the dopamine receptor. Here you see the dopamine response, and here you see what happens when you use an antagonist. No response or hardly any response, and the dopamine is sort of suppressed. For this particular case, it's a little different because the G-alpha I reduces the concentration in cyclic AMP, but it is already low in a regular resting state cell. So that's why we had first to increase cyclic AMP, and then we use the system in order to see whether it's being reduced by dopamine addition, and that only works when the antagonist is not present. What I would like to point out here again is on these 2 cases here, we have endogenous receptor expression. There is no engineering involved whatsoever. In this particular case, we used engineered cells that overexpress the dopamine receptor. That is certainly also possible, but it's not required. Now, let me get back to the histamine receptor. So here you see cells, U373 cells, human glioblastoma cells, endogenously expressing the histamine receptor. And here you see an experiment like the one I've shown you before. We monitor the impedance, we stimulate the cells with increasing concentrations of histamine. We see an increasing cell shape change. And then again, we took the impedance change here as a function of concentration, and we plotted it, got a dose response curve. This is the EC50. 0.3 micromolar. And of course, we have to compare ourselves to the established assays. So what we did is, remember the G-alpha Q couples to phospholipase C, leading to calcium mobilization. So we did what many other people do. We measured calcium mobilization. And we did this with an optical assay, a Fura assay. And what came out is this. So we got an EC50 of 1.0 micromolar compared to the 0.3 that we saw for the impedance assay. So again, very close. But there is a factor of 3 in between. And since we saw this more often, we sort of tried to see whether there was any systematics behind that. And it turned out to be true. This is a diagram here which shows you our work on HEC cells expressing the human histamine 1 receptor, so the very same receptor as I showed you before. And you see here the potency in these experiments, meaning the PEC50 in this spider diagram. And you see all kinds of established assays for this. And down here is impedance. And we used 3 different agonists, the endogenous agonist and synthetic agonist. And in this work, we always found that the potency was bigger, or the EC50 smaller, when we used the impedance readout. And we asked ourselves, is this now something that is artificial, or is there some meaning in it? And we tried to figure out what is the problem. Because what is different from this assay from everything else is there's no reagents involved. It's completely label-free. So what we did, that was a smart experiment by a postdoc who I had the pleasure to work with. She did the typical histamine experiment again. You see U373 cells stimulated with increasing concentrations of histamine and the impedance readout. And then she did on the impedance plate everything that she would have been doing when you did the Fura assay. So she applied the Fura, she applied all the reagents that are needed in order to do the Fura assay, but she did not do a fluorescence readout, but an impedance readout. And this is the answer here. So doing the very same readout impedance, but applying conditions that we knew from the Fura assay, yielded an EC50 that was slightly higher than the one that we saw there. And we have seen this reproducibly. Whenever we use reagents, the PEC50 is different compared when you study the cells completely label-free. And with this, I'd like to summarize. So the activation of metabotropic GPCRs that do not mediate any charge transfer across the membrane is accessible by electrochemical impedance monitoring, simply because you will monitor the cell shape. The sensitivity is sufficient to use cells with endogenous receptor expression. The assay can be conducted in agonist and antagonist mode, so anything that is needed to profile the pharmacology. You get the pharmacological parameters here. It's label-free, no interference from labels. Time resolution can be sort of pushed down to a second. And with the Atlas device from Nanion, we can do 6 × 96 wells. So there's really throughput. And in 1 extra minute, I'd like to show you some fun stuff that we did in the laboratory, simply to work on this quote from Ephicosthenes. You don't know what it means. That is certainly something that challenges me. And here's the experiment that we did. You probably all know about split luciferase assays. What it is, you take the G protein, and on the level of the DNA, you add part of a luciferase to it. And then you take the receptor, and you add, by genetic engineering, the missing part of the luciferase to it. So once the G protein and the receptor do not interact with each other, you don't get any luciferase activity. But once the GPCR binds to the G protein, they sort of recombine, and then you get luciferase activity. And if you add the luciferin, you will see luminescence. So our idea was, let's do this simultaneously with impedance readings. So what we did is we actually came up by wiring such a 96-well electrode array. We did this by hand. That is something for somebody who killed father and mother on the same day, I can tell you. So you see all these little wires here, and then we've put it into a TECAN reader and let it go in there. And then we did the experiment simultaneously recording the interaction of the G protein with the receptor and at the same time measuring the impedance. And this is a busy slide, but it shows you how it works. So we used the HEC 923 cells expressing the muscarinic receptor 1, and that is coupling to GQ. And we had sort of these engineered here in order to have the split luciferase. And what you see here is always the time. On the red axis, you see the bioluminescence. On here, you see the impedance. And this is for different concentrations of an agonist, which is named iperoxo, which is a synthetic derivative of carbachol. So what you see here is in black, the impedance curve. At 1, we added luciferin, and at 2, we added the iperoxo. And when you go through these concentrations, you can see that at non-nanomolar, we already see that the impedance readout responds to the stimulation, whereas the split luciferase is still rather quiet. And then you can see, you can follow yourself going to 10 nanomolar, 30 nanomolar, 100 nanomolar, 1 micromolar, 10 micromolar. So obviously for the low concentrations, the impedance is more sensitive than the split luciferase assay. However, when we traced one against the other, we learned that when it comes to high concentration, impedance saturates. Now, impedance saturates, the fluorescence or luminescence does not. And with this final experiment, which was just done recently, I'd like to stop, and I'm happy to answer your questions, not without thanking my co-workers, of course, who have done all the work, and of course the funding agency, and of course the guys from Nanion for the continuous cooperation.
Fantastic. Thanks a lot for your talk. Are there questions?
You mentioned that the impedance changes are due to the cell-based changes, right? Does it work with artificial systems like liposomes, membrane patches?
We have only studied giant liposomes on those electrodes. Of course, you can see them attaching and sort of adhering to the surface, but we never try to manipulate the shape.
Okay.
That is something that we haven't done yet.
So these impedance changes are definitely from cell-based changes.
Yes, they are from
Not
cell
because of
shape.
just binding the ligand to the.
No, of course not. No, no. You can see it from the antagonist studies.
Okay.
When you add the antagonist in higher concentrations, no response whatsoever.
Yeah, thank you very much for the inspiring presentation. You mentioned shape change, and what came immediately into my mind is the G protein G12 and G13 and platelets and shape change, because this is a dramatic change in the surface. Do you see a chance to analyze these platelets also on the Atlas system, for example?
Platelets are not really adhering, right? So this technology is limited to adherent cells, at least weakly adherent cells. And the question is whether we could come up with a strategy to get the platelets somehow to the surface. Then there might be a chance, but I can't really predict because it requires adhesion to the electrodes to do it first.
Maybe a second chance if this is for a second question. No, so thank you very much. So you mentioned the GPCRs. The majority of GPCRs is expressed in the olfactory epithelium. So this is a primary neuronal cells. Do you see a chance to attach these neurons to the Atlas system or the surface?
Well, we haven't worked with the neurons, but we have been thinking about this, of course, and we looked for cells that were genetically engineered to overexpress these receptors, and we put them on the electrodes. And of course, it does work, but we have never done it with the neurons primary cultured or something like that, only with engineered cells.
Thank you.
I maybe have a comment for you for regarding the platelets. So this is actually an experiment that we did in-house here, looking at red blood cells, coagulation of these red blood cells, and you know, getting a higher density condensement of the red blood cells. And this is indeed something that we could measure with the Atlas system, especially when we investigated the phase of the data that we capture with the system. And yeah, I'd be happy to show these experiments to you. So maybe that's interesting for you to see.
The online question, Ali? No? Can I have a question? I mean, I'm coming back to the quote, sorry. Do you really understand what it means? So, I mean, you told that that is a between the cells, basically. So if you stimulate with a hormone, so what do you think actually changes the epithelium conductance.
Why impedance goes up or down?
Yeah.
Well, that is the $100 million question, of course. And that is basically because also when you look at different cell types, there was this big dream that from the impedance profiles, from the type profiles, you can tell this is GQ, GI, or GS. We did not find this to be true because it's very much tissue specific. In one of those slides, I showed you cells where we increased cyclic AMP, and in the other example, we increased and impedance went down. But to my understanding, there are two basic mechanisms that could account for the cell shape change. First is, of course, cytoskeleton mediated. That at the end of the avalanche, there is cytoskeletal rearrangement. Second is somehow there are ion channels around, so we get back to ion channels here that are opened by nucleotides, cyclic nucleotides, by second messengers.
CMP, yes.
Yes, so it could be an indirect effect on
Phosphorylation.
the ion channels, and then we see some osmotic change. So it's basically then an osmotic change.
Yeah.
Because when you look at the time scale, this is about minutes. We measure like 20 minutes is the maximum. So this is not the direct opening of the ion channel, but it might be the osmotic change that occurs due to the disbalance that we